Right. So for my next abstract, this abstract is again based on, sorry. Yeah. So my next abstract is also based on quality assurance, but this quality assurance is primarily related with contouring for a clinical trial setting. So quality assurance of contouring for an NRG oncology protocol, RTOG 1308, which was based on author segmentation with deep active learning. Next, please. These were the disclosures of the authors. Next, please. So for precise radiotherapy for optimal treatment requires organs at risk to be accurately delineated. This is no news for us. The better the accurate the delineation, better is the plan and better is the output. However, considerable inter and intra-observer variations in the delineation of OARs have been reported. This has been studied and found. It may result in inconsistent evaluation of doses to the OARs, therefore hampering our efficacy of radiotherapy and analysis of its toxicity. Next, please. So, Low et al. This is one of the studies back in 2014, and it was published in JPO. They reported that 23% of the contours submitted to a multi-center peer-reviewed lung stereotactic radiation therapy study needed major revision. Now, this is a huge number. It is therefore essential to undertake quality assurance of OARs contouring for multi-center clinical trials because the cases come from different medical centers and so are susceptible to inconsistent contouring. Please. This QA procedure is usually performed manually by physician investigators, which is both time consuming and subjective. Again, because there will be multiple physicians involved, there will be subjective error that is instilled in this checking. There is therefore a need for an automated and objective QA method. The authors of this abstract have developed a fully automated method for the QA of OAR contour based on deep active learning. Next, please. So what they've done is that they've taken patient data set from the study, which was a gold standard atlas from the lung CT segmentation challenge back in 2017. So this had about 36 cases. Then they took 110 cases from the NRG Oncology RTOG 1308 trial. How they divide these 110 patients is going probably what we'll see in the next slide. The OARs that they studied were heart, esophagus, spinal cord, and lung, left and right. The manual contours of the gold standard atlas were drawn according to the RTOG 1106 contouring atlas guidelines. Next, please. So what were the various auto-segmentation methods that they had used? One of the most important and well-recognized methods is the convolutional neural networks, which is the CNN method. It's a deep learning method which has proved effective for semantic segmentations and is becoming a state-of-the-art method for this. But one of the biggest drawbacks for CNN is that it requires a sufficiently large set of manually annotated instances to achieve a good performance. And these manual annotations must be accurate enough to avoid misleading of the model training. So small gold standard atlas with few annotated cases is insufficient for optimally tuning these fine parameters. So we have the clinical trial data, but the data is large enough and there's a lot of variation in accuracy. So it cannot be relied upon completely. Next, please. So this is the flowchart for their QA procedure. As you can see, the authors prepared a gold standard atlas for disease type and divided the cases from the clinical trial into three major chunks. First, into the candidate set, second, into the validation set, and third, into the test set. So now we have four sets with us: the gold atlas, candidate, validation, and test. They fed the gold atlas into the CNN model. So this majorly trained the CNN model with the gold atlas training set. However, this was not sufficient enough for making a fine-tuned CNN model. So what they did was they used the candidate set. Now, how they took the candidate set is something that we'll see in a subsequent slide. But these candidate sets, about 70 in number, they were fed into the CNN model again to fine-tune it. So they evaluated the accuracy of this then attained fine-tuned model by applying it to the validation set. Now, what is a validation set? So validation set is the one which had included contours with verified accuracy. So the physicians had verified its accuracy, and then this was fed into the CNN model. The evaluation metrics included dye similarity coefficient DSC and the Hausdorff distance HD. Finally, they applied the fine-tuned CNN model and the decision criteria to the test set, which was to be tested. Next, please. And sorry, I think, can we move back to the slide? Yeah. So, yeah, let me come up to the slide. So the CNN model had demonstrated to have a state-of-the-art performance. They inputted CNN was the original 2D CT images, and its output was the corresponding segmentation probability map for the OARs. Next, please.
Dr. Sarthak, you have exceeded your time limit.
Okay, sir.
Please summarize in 1 minute.
Yes, sir. Okay, sir. Thank you. So this is, I'll be skipping these 3 slides. These are just formulas that they had used to classify their material methods. So we'll jump to the results section. Can you move to the first? Yeah, thank you. So if you can see the OARs, they had taken out the OARs and presented the result in 2 forms, the BIS and the hot-stop distance. And if you can see that all the OARs passed the passing criteria for both parameters, BIS and hot-stop distance. Next, please. So this is the error detection results table. This table lists the results of contour error detection using test set. The balance accuracy for all the OARs were greater than 0.95, which indicates that 95% of the contours were recognized correctly and no future manual rechecking was required. Next, please. So this is the image set that they had given us, and these figures represent examples of inappropriate contours detected by the QA system. The manual contours are in red and the automatically generated contours are in other colors. From the first column to the 5th column, if you can see that they are heart, esophagus, spinal cord, left lung and right lung respectively. And the system was able to identify slices with missing contours, contours drawn in red, drawn in error and inappropriate contours. Next, please. And now to summarize what the authors wanted to say was that the quality assurance of the delineation of organs at risk is essential for clinical trials in radiotherapy. They proposed an automated method for QA of organs at risk, delineation based on the active learning. The evaluation using multi-center clinical trial data demonstrated that the method can detect contour errors accurately and can thus serve an effective QA